MCQs, Sterilization Techniques in Biotechnology. Which of the following is the most reliable method of sterilization for culture media in biotechnology labs? A. Filtration. B. Dry heat sterilization. C. Autoclaving. D. Ultraviolet radiation. Answer: C. Autoclaving. Autoclaving, steam under pressure at 121 degrees Celsius, 15 psi, ensures complete destruction of all microorganisms and spores, making it the most dependable method for sterilizing media. The effectiveness of autoclaving primarily depends on A. Pressure alone. B. Temperature and time. C. Humidity alone. D. Type of material sterilized. Answer: B. Temperature and time. Sterilization effectiveness depends on maintaining 121 degrees Celsius for at least 15 to 20 minutes. Pressure only helps achieve the required temperature. Which of the following statements about dry heat sterilization is true? A. It works faster than moist heat. B. It is suitable for aqueous solutions. C. It destroys by protein oxidation. D. It uses steam under pressure. Answer: C. It destroys by protein oxidation. Dry heat causes oxidation and denaturation of cell components. It's slower than moist heat and used for materials that can't get wet. Which of the following methods is used for sterilizing antibiotics and enzymes? A. Autoclaving. B. UV radiation. C. Filtration. D. Hot air oven. Answer: C. Filtration. Antibiotics and enzymes are heat sensitive, thus, membrane filtration, 0.22 m, is used to remove microbes without heat exposure. The spore-forming bacteria used as a biological indicator for autoclave efficiency is A. Bacillus subtilis. B. Geobacillus stearothermophilus. C. Pseudomonas aeruginosa. D. E. coli. Answer: B. Geobacillus stearothermophilus. It produces highly heat-resistant spores, used to confirm proper autoclave function. Tyndallization is especially useful for A. Thermolabel solutions containing spores. B. Glassware. C. Surgical instruments. D. Metal apparatus. Answer: A. Thermolabel solutions containing spores. Tyndallization uses intermittent steaming over 3 days to destroy spores that germinate between heat cycles. In an autoclave, increasing pressure mainly helps to A. Kill spores directly. B. Increase boiling point of water. C. Produce superheated steam. D. Generate vacuum. Answer: B. Increase boiling point of water. Higher pressure increases water's boiling point, allowing sterilization at higher temperature, 121 degrees Celsius. The most effective wavelength of UV radiation for microbial destruction is A. 180 nanometers. B. 200 nanometers. C. 265 nanometers. D. 310 nanometers. Answer: C. 265 nanometers. UV radiation at 265 nanometers causes thymine dimer formation in DNA, inhibiting replication. Limitation of UV sterilization is A. Long exposure time. B. Poor penetration power. C. High energy requirement. D. Formation of heat. Answer: B. Poor penetration. UV sterilizes only surfaces because it doesn't penetrate glass, plastic, or opaque materials. Which method sterilizes plastic petri dishes and syringes effectively? A. Gamma radiation. B. Autoclaving. C. Filtration. D. Hot air oven. Answer: A. Gamma radiation. Gamma rays penetrate deep and kill microorganisms without heat, ideal for heat-sensitive materials. Ethylene oxide sterilization works by A. Protein denaturation. B. Alkylation of nucleic acids and proteins. C. Oxidation of lipids. D. Dehydration of spores. Answer: B. Alkylation of nucleic acids and ego gas reacts with NH2, COOH, and SH groups, disrupting vital biomolecules. Which of the following cannot be sterilized by autoclaving? A. Glass pipettes. B. Culture media. C. Plastic syringes. D. Surgical instruments. Answer: C. Plastic syringes. High heat in autoclaving melts plastics. Gamma radiation or ego gas is used instead. Pasteurization kills A. All microorganisms and spores. B. Only vegetative pathogenic bacteria. C. Only viruses. D. Endospores only. Answer: B. Only vegetative pathogenic bacteria. Pasteurization reduces microbial load but does not sterilize. Spores and some non-pathogenic microbes survive. HEPA filters can remove particles of size A. 0.1 m. B. 0.22 m. C. 0.3 m. D. 1 m. Answer: C. 0.3. HEPA filters capture 99.97% of particles greater than or equal to 0.3 m, maintaining sterile airflow in laminar hoods. Hydrogen peroxide sterilization acts mainly through A. Protein denaturation. B. Oxidation of cellular components. C. Alkylation. D. Dehydration. Answer: B. Oxidation of cellular components. H2O2 releases free radicals that oxidize and damage proteins, DNA, and lipids. The temperature and duration of hot air oven sterilization are A. 120 degrees Celsius for 15 minutes. B. 140 degrees Celsius for 1 hour. C. 160 to 170 degrees C for 2 to 3 HR. D. 100 degrees Celsius for 1 hour. Answer: C. 160 to 170 degrees Celsius for 2 to 3 HR. Dry heat requires high temperature and long duration to achieve complete sterilization. Which of the following can inactivate bacterial spores most efficiently? A. UV radiation. B. Alcohol. C. Steam under pressure. D. Filtration. Answer: C. Steam under pressure. Autoclaving, moist heat, effectively destroys spores, unlike UV or alcohol. What is the function of Bowie-Dick test in autoclaving? A. Detect microbial growth. B. Test vacuum efficiency and air removal. C. Measure steam pressure. D. Check media pH. Answer: B. Test vacuum efficiency and air. The Bowie-Dick test ensures proper air evacuation and steam penetration in vacuum-assisted autoclaves. The D value in sterilization refers to A. Time to kill all microorganisms. B. Time to reduce microbial population by 90%. C. Time to double microbial population. D. Temperature required for sterilization. Answer: B. Time to reduce microbial population by 90%. The decimal reduction time, D value, measures resistance of microbes to a given sterilization condition. Which of the following is not a sterilization method but a disinfection method? A. UV radiation. B. Boiling. C. Autoclaving. D. Gamma radiation. Answer: B. Boiling. Boiling kills most vegetative bacteria but not spores, hence it disinfects but doesn't sterilize. The Z value in sterilization refers to A. Temperature increase needed to reduce D value by 10-fold. B. Time required for total sterilization. C. Degree of microbial contamination. D. Logarithmic kill rate. Answer: A. Temperature increase needed to reduce D value by 10-fold. Z value indicates thermal resistance of microorganisms. Ethylene oxide sterilization is particularly suited for A. Glass instruments. B. Heat label plastics. C. Culture media. D. Liquids. Answer: B. Heat label plastics. Eco gas sterilizes without heat or moisture, ideal for plastics. The process that uses ionized gas, plasma, for sterilization works mainly by A. Alkylation. B. Oxidation and free radical formation. C. Protein coagulation. D. Dehydration. Answer: B. Oxidation and free radical formation. Plasma generates reactive species that oxidize and destroy microbial components. Which of the following sterilization indicators changes color after exposure to steam? A. Biological spore test. B. Chemical tape indicator. C. HEPA filter test. D. Bowie-Dick test. Answer: B. Chemical tape indicator. Autoclave tapes contain heat-sensitive chemicals that change color after proper exposure. Which of the following is not killed by 70% ethanol? A. Gram-negative bacteria. B. Spores. C. Gram-positive bacteria. D. Fungi. Answer: B. Spores. Ethanol kills vegetative cells but not bacterial endospores. The best method for sterilizing glass syringes is A. Autoclaving. B. Hot air oven. C. Gamma radiation. D. Ethylene oxide. Answer: B. Hot air oven. Glass can tolerate high dry heat without damage, making dry heat ideal. Which of these radiation types is non-ionizing? A. Gamma rays. B. X-rays. C. UV rays. D. Beta rays. Answer: C. UV rays. UV is non-ionizing and less penetrative, while gamma and X-rays are ionizing. The key factor in filtration sterilization is A. Pore size of the filter. B. Pressure applied. C. Duration of filtration. D. Volume filtered. Answer: A. Pore size of the filter. Microbial retention depends directly on filter pore size, usually 0.22 m. The most heat-resistant structure among the following is A. Fungal spores. B. Bacterial spores. C. Protozoan cysts. D. Mycoplasma cells. Answer: B. Bacterial spores. Spores of Bacillus and Clostridium are extremely resistant to heat. Which radiation is used in food sterilization and preservation? A. UV. B. Infrared. C. Gamma rays. D. X-rays. Answer: C. Gamma rays. Gamma radiation extends food shelf life by killing spoilage organisms without heat. What does the term aseptic refer to in biotechnology? A. Contaminated. B. Free from all forms of life. C. Containing only non-pathogenic microbes. D. Pathogen-free but not sterile. Answer: B. Free from all forms of. Aseptic means sterile, free from all living microorganisms. Formaldehyde fumigation is used to sterilize A. Media. B. Closed rooms or cabinets. C. Solutions. D. D. Powders. Answer, B. Closed rooms or cabinets. Formaldehyde vapor sterilizes large enclosed spaces effectively. Which of the following factors reduces effectiveness of UV sterilization? A. High humidity. B. Dry conditions. C. Transparent materials. D. Clean surfaces. Answer, A. High humidity. Water vapor absorbs UV radiation, reducing its germicidal efficiency. Which of the following methods can be used to sterilize culture media containing carbohydrates that are heat sensitive? A. Autoclaving. B. Tindalization. C. Filtration. D. Hot air oven. Answer, B. Tindalization. Repeated mild heating kills spores without degrading heat-sensitive media components. The F value represents A. Time required to kill a specific number of microbes at a given temperature. B. Equivalent time at 121 degrees Celsius for sterilization. C. Temperature required for 90% reduction. D. Thermal death point. Answer, B. Equivalent time at 121 degrees Celsius for sterilization. F value standardizes sterilization conditions, especially in canned food sterilization. Which of the following statements about HEPA filters is correct? A. They sterilize by chemical action. B. They rely on filtration and diffusion. C. They produce heat for sterilization. D. They work by radiation. Answer, B. They rely on filtration and diffusion. HEPA filters remove particles by interception, impaction, and diffusion, not chemical or heat action. The term thermal death time, TDT, refers to A. Time required to kill all microbes at a given temperature. B. Time required to reduce microbes by 90%. C. Temperature required to kill all microbes. D. Minimum time for spore activation. Answer, A. Time required to kill all microbes at a given temperature. TDT measures sterilization efficiency under defined thermal conditions. Which of the following oxidizing agents is used for surface sterilization? A. Hydrogen peroxide. B. Ethanol. C. Glutaraldehyde. D. Isopropanol. Answer, A. Hydrogen peroxide. H2O2 releases reactive oxygen species effective for surface disinfection. Why is alcohol used in 70% concentration for sterilization? A. 100% is too weak. B. 70% penetrates better and denatures proteins more effectively. C. 70% evaporates faster. D. 100% destroys membranes faster. Answer, B. 70% penetrates better and denatures proteins more effectively. Water aids protein denaturation. Pure alcohol coagulates surface proteins quickly, blocking deeper penetration. Which combination ensures complete sterilization of plant tissue culture media? A. Autoclaving plus filtration. B. UV radiation only. C. Boiling plus UV. D. Chemical treatment. Answer, A. Autoclaving plus filtration. Autoclaving sterilizes medium, filtration sterilizes heat label components, like hormones, added later. Thanks for watching.